Welcome, Brookings Bio students. This is Mrs. Riddell, and this slideshow is going to introduce you to the topic of DNA in Chapter 12 in your book. So you should start reading that section. And we're going to talk a little bit about DNA and RNA and look a little closer at those molecules that we learned about in Chapter 1 when we did our molecules, and we learned about carbs and proteins and nucleic acids and lipids. We're going to look a little more closely at the idea of how DNA stores information and passes that information to the cell to tell it what to do. Basically, we're connecting our genetics chapter with those molecules. How is it that genes are expressed in cells? Once we figured out that DNA was the genetic code, the race was on to figure out who's the first to figure out what DNA looked like. What's the structure? And the guys who finally figured that out are these two guys. Their names are James Watson and Francis Crick. And they published a paper in 1953 telling what they thought the structure of DNA was, and they were right. Eventually, they received the Nobel Prize for figuring out the structure of DNA. That was a pretty important milestone in science. They would not have received that award and figured out the structure without the help from this woman. Her name is Rosalind Franklin, and she worked in another laboratory. And at the time, she was taking X-ray pictures of DNA molecules and zapping them with X-rays and looking at the X-ray picture, kind of like you would look at an X-ray of your leg and see a bone inside. She was looking at X-ray pictures and trying to see the shape, the structure of the DNA molecules inside. And her pictures were shared with Watson and Crick. She didn't know it at the time. One of the gentlemen that worked in her laboratory shared her pictures with them, and her pictures helped them figure out the structure of DNA. When the Nobel was awarded, she did not receive the Nobel Prize and doesn't get credit, even though her pictures were instrumental in helping them to figure it out. And the reason is, by that time, she had already passed away. She died very young of ovarian cancer, probably caused by all the X-rays that she was exposed to during her doing her research. But Watson and Crick get credit for the DNA coming up with the structure of DNA. But Rosalind Franklin is instrumental in helping them to do that. So even though she didn't receive the Nobel, because they don't give Nobel awards to people who are deceased, her photographs, her X-ray pictures were instrumental in helping them to come up with the structure. So here's the structure. We talked about this in our very first molecule slideshow. We talked about all the different kinds of molecules. DNA is a ladder, a helix. We call it double helix. It looks like a twisted ladder with phosphates and sugars that make up the backbone, and A's and T's and G's and C's are nitrogen bases sticking out in the middle. So let's look a little closer at this molecule. If you remember from our molecule slideshow back in Chapter 1, way back in the beginning of the semester, we did a unit on molecules, and we looked at carbs and lipids and proteins and nucleic acids. And remember, this is our little building block for making nucleic acids. It has a sugar group, it has a nitrogen base group, and it has a phosphate group. And if I take these little subunits and I hook them together in a big long chain, I get a nucleic acid. The subunit we use to make nucleic acids are nucleotides. Remember that from Chapter 1. If you look back in your molecule slideshow from Chapter 1, you will see this slide. Remember, now we can make different subunits by changing what gets put in for the sugar spot and what I put in for the nitrogen base spot. And when I'm making DNA now, deoxyribonucleic acid, the sugar that I'm going to use, the name tells us, is deoxyribose. Deoxyribonucleic acid is a is a nucleic acid that contains deoxyribose sugar. The nitrogen bases that can be used in the nitrogen base spot are A for adenine, G for guanine, C for cytosine, T for thymine. We've learned those back in our molecule chapter in chapter 1. Remember, when I'm making DNA now, there's one nitrogen base that I cannot use, and that's uracil. No uracil in DNA. So we're going to put our little nucleotides together to make a chain. And remember, deoxyribonucleic acid DNA is double-stranded. There's a chain of nucleic acids that run down this side of the ladder, and there's a chain of nucleic acids that run on this side. The sides of the ladder are made out of the phosphate and sugar part of the nucleotide. Phosphates and sugars make the backbone. And that's what we kind of call the sides of the ladder. The stair steps that make the rungs of the ladder are those nitrogen bases. And remember, we said carbon is a really important atom in living things because it makes some really cool shapes in lots of different kinds of molecules. So if you look at this ring shape, each of these corners has got a little carbon atom. Carbon, 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 carbon. This nitrogen base is made up of two, it's a double ring nitrogen base. And ones that have two rings are called purines. Notice the A and the G are two-ring nitrogen bases. If it's a nitrogen base that only has one ring, carbon, 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 carbon on the corners, we call it a pyrimidine. The C and the T are pyrimidines, one-ring nitrogen bases. If you remember back in our molecule chapter, we talked about DNA and how it goes together, those stair steps. We talked about Chargaff. He was a scientist who looked at DNA in lots of different organisms. And what he discovered was it doesn't matter what the organism was or what kind of DNA they have. The amount of A always equaled the amount of T, and the amount of G always equaled the amount of C. At the time, no one really knew why that was. But now that we can see the structure of the DNA molecule, it makes sense. Because remember, when we make those stair steps, those rungs across the middle, thymine, the T, always bonds with A, adenine. And the G's, guanine, always binds with cytosine. So if you look at the ladder now, if I know what this base is over here on this side, this is adenine, what does this base over here have to be? Remember, our buddies. A's always go with T's. This has to be a thymine here. If I know that this is a cytosine, I can tell without even looking or knowing what's over here. This has got to be a G. C's always go with G's. T's always go with A's. And now we're going to, we're going to take our little ladder. And what's holding the sides of the ladder together, what's holding those nitrogen bases in the middle, are hydrogen bonds. We talked about little chemistry back in our first chapter. Hydrogen bonds are formed between the attraction of a kind of plus hydrogen on one side and a kind of minus some atom on the other side of the ladder. So there's a little hydrogen bond attraction here, kind of plus, kind of minus, that holds those nitrogen bases together. If we look now at how those nucleotide subunits hook together, remember a nucleotide is a sugar, 5-carbon sugar, 1, 2, 3, 4, 5. And in this case now we've kind of drawn the ring to show where those carbon atoms are. They're part of that sugar molecule. The nitrogen base sticks out to the side here. Those are going to make the rungs of the ladder. And this is going to be my phosphate sugar backbone. It's going to alternate between a phosphate and a sugar. And now this phosphate from this nucleotide is going to hook onto the sugar molecule of the nucleotide next door. It's going to make a long chain. If you look at the carbons, how we have numbered these carbons, 1, 2, 3, 4, 5. Notice the phosphate group is attached to what we call the number 5 carbon in that nucleotide subunit. And same with this one. 1, 2, 3, 4, 5 has a phosphate group attached to that number 5 carbon. Now this phosphate from this nucleotide is going to make a bond between the group that's coming off of this number 3 carbon on the next door neighbor nucleotide. We're going to do a dehydration synthesis. We're going to pull out a water molecule and make a bond here that's going to make my phosphate sugar backbone. If you look at how these hook together now, there's direction to the molecule. We're always lining them up and facing the same way, and always this phosphate is going to hook onto the number 3 carbon. So if you look at the chain that forms, we're going to have this phosphate at this end. This number 5 carbon is closest to this end, and this number 3 carbon is closest to this end of our strand. So we call that our 3' end. 3 with that little, like a little apostrophe, is the 3' end. This end of my molecule is the 3' end, and this end of the strand is going to be the 5' end because it's closest to that number 5 carbon. And if you look at how the strands go together now, the strands actually run in opposite directions. If you look at where those carbons are on the sugars, this is my sugar molecule. Here's my number 3 carbon. So this is the 3' end of this strand, and I'm making a sugar phosphate backbone. This end, though, the number 5 carbon here is closest to this end. This is my 5' end. If you look at the strand on the other side, the nitrogen bases are actually, and the strand is actually running backwards from the strand on this side. This is my 3' end here. This is my number 3 carbon. And this is my number 5 carbon. This is my 5' end. Notice the 3', 5' direction on this side is the opposite. We call that anti-parallel. The strands are parallel to each other, but they're running in opposite direction, and make it so that these nitrogen bases can hook together in the middle and line up correctly. So that idea of anti-parallel, the strands running in opposite directions. We're going to stop right there, and that takes us through the very first section of Chapter 12, 12-1. Stay tuned for more on DNA.